next presenter is ready. Are you?
Yes.
Okay, so we have the next presenter is going to be Dr. Anas. He had his PhD in theoretical physics. Am I right? Sort of.
So.
Yeah. From Warwick University. So it's just like what a coincidence, right? So we have a Warwick dominance in the room. And then he's undergraduate from Imperial College London. And he's currently a postdoctoral fellow with Oxford University. So so you are sort of like an oncologist, I would say. Yeah. So his presentation today, it's looking at cancer genomics.
Yes.
Right. Okay.
All right.
So Dr. Anas, take us through a maze of cancer genomics.
Assalamu alaikum everyone. So it's going to be a bit of change of pace, a lot more prettier pictures, a lot less content. Basically, as you introduced me, my background is in kind of theoretical physics or whatever you want to call it. The reason I work in oncology hopefully will become clear towards the end of the talk. But what I thought I will spend a lot of time doing today, because this topic is very involved and might be quite new to many of the people here, is I will go through some very simple introductions of the basics of what happens inside a biological cell and what is the reason why it's so complicated to deal with cancer. So just to start with, and obviously just to state it from the beginning, I won't be able to go through every single thing, so if there's something unclear, just ask me. Yeah. So just to start with, what is happening inside a biological cell on an ongoing basis continuously is that all of you know about DNA. And when you're dealing with DNA here in this kind of red thing, what you have is, if you uncoil it, unwrap it, this kind of long strand, and on this strand you have something called genes. And what there's been shown here is in this yellowish brownish color would be what you would traditionally call a gene. What we know now is that inside this traditionally called gene package are things that are actually expressed, meaning they're actually read off, and then there are kind of connecting things that are not read off, that contain other information that deal with how these bits are read off. And one gene normally contains more than one of these things that are read off. And in addition to that, across, like to the left and right of this gene, there will be things that you call promoters and enhancers, which are not shown here, but like promoters and enhancers, which either tell you to read up more of this thing or less of this thing. So actually it's a very complicated picture. If you look back even a few years, none of this was known, and people assumed that all of this to the left and right of this gene is junk. All of these things in the middle are junk, but this picture is continuously changing. So even in 5 years' time, this picture might be out of date. Anyway, so what you have is when this kind of reading mechanism inside a cell that reads off information that is then transformed into protein would look something like this. This is obviously still a schematic, but it would look something like this, that you have a promoter region that is binding to this kind of protein structure, and that promoter region is binding here. Then you have also some kind of enhancer region binding, some kind of enhancer region binding, region on the DNA binding at another place, and then you have this kind of gene thing here. And this construct happens every single time you read off a section. what then happens is a new protein, shown here in green, comes along, pulls apart these two different strands of DNA, and starts reading off only one of those strands. So it's even this kind of simple process starts to look a bit complicated. But another thing comes into it, right? This is what you would call a chromosome. The human body has, I'm going to say it wrong, I'm going to remember it wrong. Biologists in the room, how many? 23 chromosomes in your body. But what a chromosome is made of is, if you start to uncoil it, it's made up of this kind of strand that looks fairly uniform on a coarse-grained scale. If you dig a bit deeper into it, you have these things called nucleosomes. Basically what it is that a lot of the DNA is highly charged, just because of the way it's made up. These nucleosomes are neutral things that allow you to wrap it very tightly together. You have like a massively long thing that you're wrapping 23 of these cramming 23 of these into the nucleus of a cell. So the way this is achieved is with these neutrally charged nucleosomes around which they wrap. In addition to these nucleosomes, you have something called histones. Don't forget what they're called, but what they're about is, when I was showing you this earlier picture of things being read off, these histones will allow you or not allow you to unwrap certain sections on a chromosome. And what that allows you to have is multiple cells in your body that behave in completely different ways, but are made up of the same building blocks. So every single cell in your body has the same building blocks, but due to things called histones, different building blocks are active in different cells in different combinations, right? But in addition to those kind of histones, we also have something else, which is methylations. Yes?
I just wanted to clarify on this. Does that indicate that the histones themselves are different in the different cells?
Yes.
Behave differently?
Yes. Yes.
Okay. So everything else is the same, but the histones are different in different cells?
Yes. Yes.
So what causes that to be different in different cells?
We'll get to that.
Okay.
Are histones proteins? Sorry.
Yeah, they're also protein. Pretty much everything that happens in a cell is made up of proteins. I mean, it's a simplistic view, but yeah, pretty much. Then you have additionally, on top of these kind of things which activate and inactivate things, we also have things called methylation groups, which attach to different parts of your DNA and mutate or activate it. So that's an additional thing. And all of this kind of framework and some other things which I'm not going to go into make up what is called the epigenome. So you have this kind of complicated mechanism of reading up information from the DNA, and then you have this epigenome which adds another layer of complexity to this reading information from the DNA. This kind of picture, so the kind of DNA, the kind of genes you have in your system, does not change significantly over your lifetime. But the epigenetic information changes continuously throughout your lifetime. And it's influenced by things like your environment, things like your diet, things like, you're out in the sun, it changes, a whole host of things. Even emotions can change these things. But it's still a very open area of research. We know little bits about it, but not full picture yet. So the reason I'm talking about these things is to give you an appreciation of what's going on in the cell and why it's so complicated.
Sorry, just to maybe simplify a bit, and the reason these things are changing is because the histones are changing?
No, the histones and other things. So one of the other things is these methylations and histones and other things as well, which I'm not going to go into. It's a long story. So now we come to what I do is cancer. What is cancer? Simply speaking, it's a disease which is due to an uncontrolled proliferation of a cell. That's, in simple words, that's what it is. You've got some cells which keep multiplying, keep repeating, and then you've got tumor to cancer, right? But what people have done in the past 15 years or so is try to come up with a picture that defines what cancer is. And me coming with a background in physics, I think this picture is so descriptive that it might as well not be there. But anyway, but it gives you like indications of what things can go wrong in a cell for it to lead to a cancer. And the important thing to remember here is a single one of these things going wrong does not create a cancer, but you have to have multiple things going wrong for it to happen. So for instance, one of the things is you have something in each cell called proliferative signaling, and that's about making your cells grow, proliferate. We need that in our cells because as we grow, as our cells die, we need to replace them with new ones. But cancers utilize this and, not utilize, but misuse this to start proliferating without end. But our cells have mechanisms against it to prevent it, and if it's happening too much, to kill that cell off. That happens a lot. But then those cancer cells can have mechanisms to evade those growth suppressors. Next stage is in a cancer is an activation of invasion and metastasis. What that means is you having cancer in one specific place is not dangerous. It's when that cancer starts spreading throughout your body, that's when it becomes dangerous. That's when it becomes untreatable, or difficult to treat rather. Then you have something, so we know that we eventually die. And one of the reasons is that our cells, if we don't get a disease, is that our cells, all of our cells have a limited lifetime. When you read off these genes that I was talking about earlier, at the end of each of them is something called the telomerase. It has a fixed length, and as cells duplicate, that length starts getting smaller and smaller and smaller. When that length has disappeared, your cells cannot replicate anymore. So it's like an inbuilt end of time clock in your body. But what these cancer cells do is evade that. They increase the length of the telomerase and many other things. Inducing angiogenesis, which is for something to keep proliferating in your body, it needs blood. It needs the nutrients in the blood. So what cancer cells have developed is they replicate the signaling mechanism of your body and start calling for more blood vessels to be built into that region to get more nutrients. Which sounds a bit crazy, but yeah, that's it. And resisting cell death. The cell has many checkpoints which will kill off the cell if it does any of these dangerous things. But cancer cells have got an inbuilt thing where they start evading those kind of death signals. And that's one of the reasons why studying cancer is so incredibly complicated. Now talking about types of cancer. Some of you may know this, but I thought it's interesting to put this up there. Two of the most prevalent cancers in the world at the moment are breast and lung. So this picture shows you the length of this kind of strip, shows you how prevalent they are. The size of this kind of bar on top of that thing tells you how many of those types we can cure at this stage. And the blue is for female and the red is for male. And the things on top here tell you what are some of the causes for it. So breast cancer, for instance, which is one of the most dangerous diseases for women, is caused by being overweight, drinking too much alcohol, by the occupation you have, some kind of genetic defects, inactivity, and not breastfeeding. So this is something that I found very kind of, it's sad, but in a sense, I found beauty in the teachings of Islam here, because Islam teaches us that having kids is part of your duty as a human being. And what we found was when they did a research project on about, I think, 100,000 or 200,000 patients across Europe, female breast cancer patients, and what they found was a common cause for catching breast cancer, and in fact a way of reducing the risk of breast cancer is having kids. Having kids before the age of 35 significantly reduces your risk of getting breast cancer. Having a second kid reduces even further. Breastfeeding reduces your risk even further. So all of these things that are kind of, inbuilt in our nature and that Islam teaches us to follow, reduce our risk of getting illnesses. And I think that's, strangely interesting. Another thing that you might have already known and guessed at is lung cancer. Biggest cause is tobacco, smoking. And the risks are just phenomenal. Smoking is just increases your risk dramatically. But obviously, and not only here, but you will see tobacco is a cause in bowel cancer, in bladder cancer, kidney cancer, loads of cancers. Tobacco is a big cancer genetic cause. But yeah, I just thought it was interesting to see.
Nice infographic.
It's not mine. But why, in addition to all these things that I talked about, that we have cured a lot of these, why is it so hard? Because going back to that picture that I had up earlier, and I think this is a slightly more advanced version, but anyways, for many of these things, what we have found is different things that cure them. So we have got some kind of thing here, EGFR inhibitor, that this proliferation signaling that I was talking about, that stops it, many of the others as well. But the issue is that these kind of signaling procedures, or these kind of things that allow us to kill a cancer cell, these things are used in healthy cells as well. So actually when you're undergoing chemotherapy, what you're doing is pumping poison into your body. And that's why you see these harsh effects of chemo treatments. And this is an issue that we're trying to deal with. And that's why the aim is to make these kind of drugs as specifically targeted to the cancer cell as possible. But because they use the same mechanism as other cells, it's difficult to do. Another thing is, this is a kind of schematic of one simple signaling thing which deals with the estrogen receptor and PGR activation, which, if I remember correctly, is to do with cell death, maybe? I don't remember, sorry. I don't remember. But it's basically one of the common mechanisms that are prevalent in many cancer cells. What we have found is that we found a way of inhibiting one area of this pathway. So for instance, you get some kind of signal from the outside of the cell that kicks off all these downstream effects that lead to some kind of effect on your genes here at the bottom. Now, what we found is that one of these areas is mutated, one of these proteins is mutated, so if we latch on that specific mutation, then you get changes in that specific area. Then you can targeted these cancer cells. But we started using that kind of treatment. What we found a few months later, the cell decided, all right, I can't use this anymore, I'll go around here. So that's one common issue with cancer cells. They're highly resistant to these kind of breakages because they found ways around it. This is one of the simpler pathways, and you already have like multiple routes to get down to this mechanism that does something at the bottom of the cell. So that's why attacking it at one place is sometimes not sufficient. That's one of the issues. Another issue is the heterogeneity of one single tumor. So you have one single tumor, but it is made up of multiple different subtypes. So you apply a treatment one to this kind of heterogeneous system, and what you end up getting is this light blue thing, and then you get lots of these dark blue things, which are made up again of multiple things. You have a second treatment, and suddenly you get these other ones surviving, and it's still there. The cancer is still there, and it starts proliferating, and you still form these metastases throughout your body, which are hard to cure. So these are kind of, I just brought these up to highlight some of the issues that you deal with trying to cure cancer, kind of. Now I come to a bit of the work I am doing personally. I'm almost done. So,
don't worry, you've got,
all
we've
right.
still got about 7 minutes.
Oh, that's perfect.
Yeah,
That's
so
perfect.
keep going.
So systems biology, so I'm officially employed as what they call a systems biologist, whatever that means. But here is one possible definition of system biology, is like a computational and mathematical modeling approach to understand biological systems. And it's a very interdisciplinary field, as in I work with a lot of different people from different areas, a lot of biologists, and it focuses on complex interactions within biological systems using a histological approach. And what I mean by that, holistic approach, sorry. What I mean by that is biology is based on a reductionist system. So you try, every science is based on a reductionist system. You try to reduce the system to its simplest components and say, okay, what can we do with this? How can we understand this? And this approach is you forget about the reduction and you look at the system as is and try to gain some understanding out of it. And obviously I can't talk about all the projects I'm working on, so I decided to focus on one of them. And this is like a general approach that we have in biology, is that you have some information of many different cancer types, some kind of information about patients, and you have some kind of information, so like cancer and patient information, and then you have information about each patient, what is happening on their DNA, do they have mutations in their genes, like changes in their genes. These copy number is another word for like specific changes on the genome. Do they have these different methylations that I was talking about earlier? Lots of other, types of data that we have. And gene expression as well. And what you try to do is take all that information, take it together with different tumor types, and try to construct one coherent picture that will either allow you to catch the disease early, because that's the game in cancer. If you catch the disease early, there are ways of curing it. If you don't catch it early enough, even the most curable cancer becomes uncurable. Because the types of ways that you would cure a cancer is, as I was saying, pumping poison into your body. If you do it targeted enough, you can kill a tumor and you're fine. If you don't do it targeted enough and it's all over your body, there's nothing you can do. Radiotherapy is another way of curing cancer, but again, it's dangerous to the cell. And in fact, there are many cases where due to radiotherapy, patients died. So you have to make it very, very targeted, but as soon as it's too late and the cancer is spread throughout your body, you can't do anything. So these are kind of the things you deal with. And that's why catching the cancer early is very important. And sometimes you use these kind of methods to understand the cancer better, to then devise methods of catching the cancer early. So one of the projects I'm working on deals with some parts of this data, starting with gene expression. And the idea is this, that I was telling you that you have different pieces of these exons, these kind of dark orange things in one single gene. So what they have found recently is that in some cancer types, if you have only one of these active, the outcome for the patients will be different than when you have, two of these active, for instance. So the idea is this, that if you have this, red subtype, where these two are combined together to form a gene and then a protein, then the patient might die, but if you have this one, the blue one active, the patient might live. So these are ways of distinguishing the patient and also making the treatment targeted. So my approach is looking at gene expression data, which looks at how much of this stuff is read off, and combining it with methylation of the kind of region to the left and maybe also to the right of this gene, combining these two data types to understand and make a genome level approach to identify which of these genes has got this characteristic for a specific cancer type. And that will allow you to catch the disease very early on in the clinic. And this would be, this is one of the approaches that you could use, that you go into a hospital, they will do a screening, they would check, do you have this particular subtype, then you need to quickly get this medication. Or you don't have it, you need to take a different approach. So this is, simply speaking, this is one of the projects that I'm working on. Right, so two of the people I'm working with on this, some of the people who give me money, and not all places in Oxford have got fancy old buildings. So yeah, thank you for listening. Any questions?
Right on time. Okay, thank you very much. But a bit of, sorry we didn't start with a bit of housekeeping exercise.
Yeah.
If you can, just turn off your phones all day, or you just put them on silent mode so you don't have distractions while the presentations are ongoing. Well, there are not really any fire regulations, so I know if there's any sound, it's a live fire, and we have to get through the earliest exit as possible. All right, so, yeah.
Generally, if you cure one type of cancer, it appears in other part of your body. So in that way, it means that the information is much more deeper. There's something, the whole is much more deeper.
No, it can be as simple as this, that, as I was saying, the same, so if you have, like, say, lung cancer, you might not notice, but a small chunk of that cancer might have broken off and traveled via your bloodstream to a distant part of your body and started proliferating there. So you treat that lung cancer because it's big enough for you to catch, and this is another issue. Sometimes if it's too small, you can't catch it. So you catch it there and you cure it there, but that piece is already somewhere else. And yeah, but there are also aspects, and this is one of my other projects, where the issue is much deeper. It's something, they have fancy words for this, but something they call chromosome shattering. So you have chromosome shattering events happening due to external causes, which mean that the chromosome just almost explodes and is rebuilt wrongly, and that leads to issues. So these things happen as well.
Yeah.
So you mentioned about what was the thing at the end of the gene phenome?
Telomerase.
Telomerase. They said that each time a device they sort of shrink and that sort of. Is there like some sort of actual relation that does it half or does it just reduce or?
I don't remember. I think there is, but I don't remember. In fact, there is an interesting piece of research on this recently. What they've done is, I don't remember where, but they have basically artificially lengthened these telomerases in mice and increased their lifetime by 6 months, which, if you know about mice lifetime, it's an insane amount of length. You've got something to add, I assume.
Just 1 second, I think it's telomerase, not telomerase.
Sorry, telomerase. Telomerase is the mechanism.
Enzyme, which is actually
Yeah.
breaking down.
Yeah. Telomerase, yeah, sorry. Yeah, exactly.
All right.
Yeah, there was another question, but I'll just think it up.
But how advanced is it to curing cancer? In terms of when it's Because I mean, this is the same old story here. When it reaches, it's like a very later stages.
Right. Let me
It's
just pull
incurable.
up that slide,
Yeah.
if I may.
But
Right.
I mean,
So,
there's
for instance, what
a lot of news out there that's
Right.
research has been ongoing as
Yeah. So the incredibly complicated thing, and again, it's such a vast subject, so I didn't get into it.
Yeah.
Each of these cancer types is completely different. When I'm studying them on a genomic scale, they don't compare at all. So each of these cancer types has to be treated separately.
Okay.
Treatments have to be devised separately. So that's what you can see here. Breast cancer is maybe a third of the types of breast cancer. And each of these breast cancers each of these cancers consists of many different subtypes, which can't be cured with the same thing. So when you read in the newspaper that they found a cure, it's probably one of the small subtypes somewhere in the cancer. So this shows you, for instance, it's only 1/3 of the breast cancers we can cure, many of the lung cancers, many of the bowel cancers, none of the prostate cancers, things like that.
You didn't talk about blood cancer.
Yeah.
Quite. Is it in a different league altogether?
Yeah.
Okay.
Yeah. So it's complicated answer. Because there is no one mechanism,
Yeah.
there is no one way to attack it. So for instance, if you get, AIDS, another big disease,
Yeah.
you know exactly what is the root cause.
Yeah.
So you can, and the root cause is very specific. It's that one virus.
Yeah.
So if you find a way of attacking that virus, you've cured AIDS. Cancer is not like that. Cancer is a disease of your own body, of your own body's making. So there is no external host that you can attack.
Okay.
So that's why it makes it complicated.
Okay. All right. Yeah.
If you could go to that picture, the one with your project.
Oh yeah, okay.
Yeah. So yeah, that's just I think the one previously before that one. Even this one is fine, I think.
Yeah, okay.
So this is something which I found very exciting as well.
Yeah.
Something new for us as well. In regards to just the image below, what
This-
could we see? Yeah,
this one here?
here.
Oh,
So-
I don't know what exactly this project was, but it's basically, I mean, this is something I create as well. I don't create the images, but anyway, but it's the idea that you have each of our genes don't work independently, so each of our genes forms a complete complex of things. So what you do is, once you have you incorporate many different data types, and what you try to create is a picture of what is happening in that particular cell type based on all of the different connections of the genes. So each of these nodes, each of these points will represent a gene, and each of these lines will represent a connection between these genes. So the idea is this, that if you create a picture like this, what you should be able to do is identify something that is specific for that particular cancer type that is important for it to survive, and you can attack it. Something like that.
And, okay.
So, in I guess a simplistic way of mine's understanding of what you've said as to what you do with the data, then you've got all of these different information about genes, or it's just to see that you've got different data on a patient, and you're trying to get some statistical correlation between them to identify some patterns that you could then use as like markers or signals.
Well, yeah, but it's not quite. It's not quite just simply a correlation, but it's a.
Because you said that if we know that then if we identify that in a patient earlier, we could also be, rather than relying on traditional methods of seeing a tumor and then knowing that there's a cancer, we could actually know upfront by looking for those things if we know that those things happen in cancer. But does that, is that not based on an implicit assumption? Maybe that is a true assumption that these patterns that you're looking for, they don't change. So could it be that these patterns develop because there's cancer, before the cancer those patterns weren't there, there were some other patterns. So do we also have like a time factor in these things that for those patients?
Definitely, over time things change and things.
But do we get that as well with these things, or are these things for the time being only
No,
for
this
patients
will
that
be
you have?
only that time. And this is one of the issues that we were talking about yesterday night. The blind prediction in biology is very, very hard. because my work is based on this kind of data on a cancer, I can only make predictions on cancer. I can't, because it doesn't carry, so there's no like underlying mathematical description that would allow me to say that if I identify these parameters, then if I perturb the system, this is going to happen. Maths and physics are like these, you know, I'm going to get nostalgic, but is one of these, you know, the beauty of those is that you have this incredibly complex substructure that gives you a description of the whole subject. And you can go ahead, when you find like something interesting, you can go ahead and say, what happens if I add this perturbation? And then you can theoretically say, this is what will happen. And because of that, I should observe this. And then you can go ahead and check it. That's why theory and experiments work hand in hand in physics. But we don't have this kind of underlying theoretical structure in biology. A lot of these, you know, even the hallmarks of cancer, the kind of big circular picture that I was showing earlier, is kind of an idea of say, oh, this is what's happening. But the reason why I find that unsatisfactory is because I'm used to physics where it actually describes things. But this is just an observation.
Yeah.
So it's not, yeah.
So,
does that answer your question?
yeah, I mean, I guess it's lead on, but I will.
Yeah.
That was the same question I was.
Yeah.
Yeah.
So, because I guess sort of the lead on from that is that doesn't mean that in terms of the work you're doing, is it outcome simply to then identify some patterns of cancers to see how we can target those cancers rather than saying that these are the patterns which if we identify in normal or relatively healthier human beings early on, we can actually predict the fact that there'll be cancer. So that's not what we're then looking at.
So no, for me personally, what the ideal situation will be, because, you know, it's going to sound a bit crass, but I don't particularly care about a specific cancer type, or I care about the method. And what I want to do is come up with a method that would allow you, given some data, to identify these kind of patterns for any kind of cancer type.
Yeah.
So that would allow you to come up with a signature for any cancer type that you would look for.
But
that's, you know,
for
that's...
diagnostic purpose. For diagnostic purpose to
For
identify
diagnostic purpose.
earlier.
Yeah, to
But-
catch it early. But that would be, you know, I have to add this caveat that I'm not expecting that to happen completely in all possible, without loss of generality, because that's not going to happen. Yeah. Yeah.
I think the only issue I would have with this is in regards to cost.
So from my side, there's no cost involved. I'm just looking at
Everything's on
doing some number crunch. But yeah.
All right, we've got to the end of the presentation.